I wanted to explain about protoplast. So protoplast can be generated by stripping the cell wall from plant bacterial cells or fungal cells. So there are different type of cells like plant cells, fungal cells or bacterial cells, right? So these from these cells the protoplast can be easily obtained. So what is protoplast actually? The cells which doesn't contains cell wall is called as protoplast. So how we are going to obtain these protoplasts? Let us see now. So let's say there are different type of cells which I have mentioned you just now. They are plant cells, bacterial cells and fungal cells. So in the case of plant cells, once it is treated with enzymes called as cellulase, pectinase and xylanase, then what will happen? Then the cell wall of this plant cell will get ruptured. Once the cell wall of the plant cell will get ruptured, then the protoplast which is present inside that cell can be easily obtained. It can be easily exposed, right? In the same way, these bacterial cells, once it is treated with enzymes called lysozyme or else lysostaffin, then what will happen? Then the cell wall will be ruptured and the protoplast which is present inside this bacterial cell can be easily obtained. In the same way, fungal cells, once it is exposed to the enzyme called as chitinase, then what will happen? Then the cell wall of this fungal cell will be ruptured and the protoplast which is present inside this can be easily obtained, right? So in this way the protoplasts can be obtained, right? So this is about just the protoplast introduction which I wanted to tell you about. And here now let us discuss about protoplast culture in detail. So there is a procedure which is mainly present behind this protoplast culture. So understand, okay? So it is very easy. So firstly what you are going to do, you are going to select the explant. So what is the explant which you are going to choose? Leaf, because here we are going to perform the experiment called protoplast culture. So protoplasts are obtained from the leaf, hence we are going to take the example of leaf as an explant in this experiment. So now what you are going to do, you are going to take this leaf and you are going to sterilize it. So the process of the process of sterilizing this leaf is called as sterilization. So how you are going to perform the sterilization process? See, firstly you are going to take that explant. Explant is nothing but the leaf which we have already chosen. And now this explant should wash under running tap water.